Hi, welcome. Just setting up right now. What I need to do. What am I doing? It's a good question. What am I doing? Do you have any questions? Have you got topic areas you'd like to cover? I am ready to answer away if you have any. Um, dexterity. Are you able to go over biotechnology? Sure. What bit of biotechnology would you like me to cover? Go. Oh, honestly, I feel like, well, it's one of those days where it's so hot that you just don't know what you're doing, whether you're coming or going. But here we go. So I am on my YouTube channel if you want to come across. There is a link to the Zoom as well. So yeah, please do come and join me. And yeah, so you were saying about biotechnology. So my question to you, Brie, is which part of biotechnology are you struggling with the most? Because obviously biotechnology's got quite a big part to it. Is it the fermentation side of things? Is it the aseptic technique side of things? Is it the genetic engineering? What part of it is it that you're actually looking for? Amna, of course I can. Hold on, let me get the paper up. Um, so you want 2017. Right, let me think this one through. Right, 2017. Have I got this one open already? 2017. What paper did you say? Paper two. Let's see. 17. And you want me to go through question 18 BII, question 19, question 18. So is that the tissue culture one, Amna? Just to double check I've got the right one. So the one about tissue culture. Hi, Alex. Yeah, of course, Alex. Send me through um, the question. Let me know where it's from, and I'll do this. Please, can you explain how to do serial dilutions? Um, yes, I can do serial dilutions. I will do that after. I've got one question that Amna's asking me to look at with regards to the question. No, it's not tissue culture. Oh, have I got the wrong paper? Hold on, let me have a look. 2017, paper two, you said, right? 2017, paper two, 18 BII, 18. Hold on, I'm going to share this with you. Okay, you were thinking I was going insane there. Right, okay, that's fine. So we want to go, so let's go to this paper first, work our way through this paper, and then I will answer the question that you have got. Would you be able to go over how to search this? For example, how many organs would yes, I will do. So Naveen, I will do that one after I've answered this question. So it's just it should be coming up on my screen in a minute on my YouTube channel. For those of you who are in Zoom, you should be able to see the question right now. So you're saying on the mark scheme, let me just get this one up. It's from paper two, 2017. And it is question 18B. Oh, went too far. Let's see what it is saying on 18B. So 18 BII. State how a clone of potatoes can be produced. Right, so you're asking for a clone. So the fact that we know that it's going to be clone, we know we're going to have to do tissue culture. So you're saying why ignore pathogens in the marks game? So why is it saying ignore pathogens in the marks? So let me just look at this one. Because it reduces the microorganisms. Well, because you're thinking about pathogens. When you're saying pathogens, pathogens are only organisms that are going to be carrying a communicable disease. Whereas if you want to reduce microorganisms, they might not be pathogens. And that's why, because a pathogen could be a virus, a pathogen could be a protoxistin, it could be a fungi, it could be a bacterial. So the fact that they're saying specifically to ignore pathogens, because what they're saying is they don't think you know that a pathogen could refer to any of those four things. So that's why they're saying you're not giving enough. Whereas if you say microorganism, then that would be the case for that one. Okay, so which one? 19B2. The zero dilution question I will come to, and Alex, I will look at yours in a second. Let me just finish this one. 19B right. Okay, so the mutation. Oh, I hate this question. I hate this question. If I'm honest with you, Amna, I don't like it at all. Because this one's a weird one with the ranges that it wants you to have and how you work it out. So in this one, right, so we got to look at the mutation rate in the mammals, and it can vary as much. So you're told it's as much as 20% between the species. And then you're asked to calculate the time since the phylogeny of humans diverged from the chimpanzees and the range over which these estimates may vary. So in this case, when we're looking at this answer, and honestly, I hate this question because there are always going to be some questions for us as teachers where we just look at it and go, what were they thinking when they wrote this? And some people who love the maths questions and think, do you know what, that's so easy. But for me, I'm like, what are you giving me? Why are you doing this to me? You're making my life horrendous right now by giving me these parts. So when you look at the graph and you see that they want you to note that the number was just, we've got it 5.25 plus or minus 0.25 million years. So you've got 5.25 plus or minus 0.25 million years, and your range is 4.2 plus or minus 0.2 to 6.3 plus or minus 0.3 million years. So that's obviously what's in the mark scheme. And so you're asking as to where did the 0.25. So this time since divergence, oh, gone too far, hold on. So the time since divergence, so they want to know about humans diverged from the chimpanzees. So the time taken for the humans to diverge from the chimpanzees, and they want you to note. So if we're looking at this, and I zoom in a bit. So what you've got is these numbers. So you've got 5, you've got 6, you've got 7, you've got 8, you've got 9. So they wanted you to say that it was 5.25. If we're looking at that scale, so here we can tell that that's definitely one of the millions of years, right? And we are just only slightly past, we're not at halfway, we're not at the end, we're only part of it. And so therefore, that's where they want you to get that you are just outside of the 5. So that's why it's an estimate then it would be 5.25, and you're actually saying that the error in your data is that point there. You can be sure up to 5, but you can't be sure of the 0.25. So what you're saying is it may be 5.25, but it could actually be 5.5, or it may be back to the 4.75. So you're basically saying plus or minus that 0.25. Oh, I just added in an extra 25. That was silly of me. So this is why I'm getting tired. 5. Try that. Maths and me. There we go. So we're saying it's down to 5, or we're saying it's up to 5.5. We're saying we're definitely, we think it's 5.25, but there is uncertainty in our data. It's a horrible question. It's one of the worst ones I've ever seen. I've never seen a question quite as rough as this again. So Amanda, does that help you on how to do that one? But it's a horrible question. I never like this question at all. Whenever I've seen it and done it with my students. Right. Hold on one second. So Alex, let me have a quick read of yours, then I'll do the serialization. So the question is, identify one difference between prophase in mitosis and prophase 2 in meiosis. Okay, so in prophase in mitosis, you know that your chromosomes are condensing, you know your nuclear envelope is disappearing, you know that your nucleolus has disappeared in prophase. In prophase 2 of meiosis, you've already gone from a diploid cell to a haploid cell. And so because of that, you've already now only got one of the chromosomes for each of the chromosomes. So you're now down to haploid. So 1 mark, and I said in meiosis, prophase 2, homologous chromosomes. No. In meiosis, prophase 1, Alex, you got that confused. Homologous chromosomes pair to form bivalence in prophase 1. When they get to prophase 2, you've already gone from a diploid to haploid cell. So I hope that helps you on that one. Right. Please, can you do GCSE as well? So what do you write? So what's the difference? The difference is that the chromosomes that are condensing only have one of each of the chromosomes because they are now haploid rather than the fact that they're diploid. So in prophase in mitosis and prophase 2 in meiosis, that's the only difference that it would be looking for you to state. Oh, second. Right, so hold on one second, the range, as to the range of where it's going to go from the, and the range over which these estimates may vary. So this is where, to be honest, I'm not going to lie to you, I'm not entirely sure how to finish that answer. Because that actually is a case of what they're saying is you're going to go all the way down to 4 and then all the way up to 6, but I'm not sure where they're going to get. I can understand if they said the range was from 4 to 6 because you're saying it's between there, but I don't know where they've got the 0.2 from and where they've got the 0.3. So I wouldn't want to give you the wrong way of working it out. Let me just quickly check if the examiner's report actually says anything specific to it, because half the time even that doesn't give you information. I don't have it there, but if anyone knows the answer to this question, is a good mathematician, please feel free to shout out. Please go over OCR paper 2 2019 question 5. Yes, I can, because I've got 2017, haven't I? Give me 1 second. What I'm going to do is I'm going to first of all do a little bit on the serodilation because I have been asked about that one. So give me a second. Let me do this on serodilation. Now, I did a question today. Let me think when I did that question, if I can get it up for you, which was a good one on this. Um, this one. Let's see. I'm going to. Right. So the question was asking about. Okay, here we go. So I'm going to change the numbers slightly to make it a little bit different, but the same. So in the question that I was looking at today, and I'll be careful because obviously I'm not allowed to share things which were 2024, but it was very similar when it came to zero dilution. So if you have a zero dilution, so let's say I have a conical flask, and in that conical flask, I have got a bacterial culture, and that bacterial culture, let's say, is 20 centimeters cubed. Okay, so I've got 20 centimeters cubed. It is the volume of my bacterial colony, and we're going to say that this is Staphylococcus aureus. I'm just going to give a number here. So I've got Staphylococcus aureus. I've got 20 centimeters cubed of this bacterial culture. Now, what I would say to you is, I would say that when you are doing this question, I would now do it this way because this makes sense to me. So I would write a little table that says cells and says volume. Okay. So this is a slightly different way to how I've done it before, but I feel like this works for me now. So I'm going to show you this way. So we know in 20 centimeters cubed, we have got a set number of cells. We're not sure how many cells are in that 20 centimeters cubed, but we know we've got 20 centimeters cubed. So what I'm going to do is I am going to take 10 centimeters cubed from my 20 centimeters cubed, and I am going to put that into a new test tube. So I've now got 10 centimeters cubed here. So this is what I've done. I've taken 10 centimeters cubed out of that. So whatever is in my 20 centimeters cubed, I'm taking 10 centimeters cubed of that. So I am going to divide this to get to my 10 centimeters cubed. I'm going to divide this by 2. So whatever number was there is also going to need to be divided by 2. Okay. So now what I'm going to do is now I'm going to tell you that we are going to take out 1 centimeter cubed out of this 10 centimeter cubed, and I am going to add in 9 centimeters cubed of distilled water. Now, therefore, in this case, I now have 10 centimeters cubed. I'm going to do the same again. I'm going to take out 1, excuse me, 1 centimeters cubed. I'm going to add in 9 centimeters cubed. And then what I'm going to do is I'm going to take out 1 centimeters cubed, and I'm going to plate that 1 centimeter cubed. Okay. So if I do this, then what I'm going to do is I have 1, 2. I've taken 1 centimeter cubed here. I've added in 9 centimeters cubed. So I have 10 centimeters cubed in there. I've then taken 1 centimeter cubed out of here. I've added in 9 centimeters cubed here. I have 10 centimeters cubed. Okay, so that's what I've done. So if I were to number these, this is number 1, there's number 1, this is number 2, there's number 2, there's number 3, which is here. Now what I'm going to do is I'm going to take out 1 centimeter cubed, and I'm going to put it onto a plate. I'm going to spread it out, and if I spread it out, I'm going to count the number of bacterial colonies that I actually am able to separate out and visibly see. So if I count this out, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So 2, 4, 6, 8, 10. So I have got 10 cells, because I've got 10 colonies. I've got 10 cells in my 1 centimeter cubed that was plated, okay? So I'm hoping that you are following me up to this point as to where I have got to. Don't know where you can see and if you are there, but let's hope you are. So if I have 10 cells in my 1 centimeter cubed, which has been plated, what I'm going to have to do is go backwards. So what I did was, out of my 10 centimeters cubed, to get that to 1 centimeter cubed, I divided that by 10 to get 1. So therefore, because I divided that by 10, to go the opposite way, I'm going to times my cells by 10. So now I've got 100 cells in number 3. And then I'm going to do the same again. I needed to divide that by 10 to get to my 1 centimeter cube. So to go back again, I'm going to times that by 10, and that's therefore going to get me to 1,000. Same again, to get from my 10 centimeter cubed to my 1, I divided it by 10. So I'm going to times it by 10. And when I times it by 10, I've now got 10,000 cells in the first tube. Well, remember what I had to do was I actually divided it by 2 to get 10 centimeters cubed of the original. So therefore, if I'm going to go backwards, I'm going to times it by 2. And that therefore means that I would have 20,000 cells in my original 20 centimeters cubed. So by counting backwards, I've worked out that there were 20,000 cells in the original, which had 20 centimeters cubed. So why is the last one divided by 10? Because I only plated out 1 centimeter cubed and I had 10 centimeters cubed in there. Does that answer your question? So it's important that you realize, and that's why I find actually this table process works for me and it makes it make sense. And I never thought I would say this, because when Alan started doing this on a table, because I was struggling with it because I was drawing my diagrams and I was like, oh, where am I going back and forth, back and forth? And I was getting myself confused. I was tripping myself up. And so what I was doing wrong is I was forgetting that when I'm removing a sample, that sample was in that 1 centimeter cube. I wasn't changing the number of cells. So that's what's really important. Oh, is it? There we go. Oh, how did your exam go today? Oh, you're very welcome. I hope that has helped on that question. Right. Somebody said, hold on 1 second. It was 2019. Abira, I'm not sure if you're still in here, but if you are, you want me to do 2019, paper 2, question 5. Right, let me get it up. 2019, question, paper 2, right, paper 2, 2019. Hold on. And then I will look at the next questions that have been listed. Right, so let me just, where do I want to go here? Right, 2019. Oh, crumbs. Sorry, I've just realized people are waiting to join. Sorry, guys, if we're waiting to join for ages there. I didn't actually notice. My bad. Right. What am I doing? Question 5. Ah, love this question. Okay, so Abira, if you are still in here, then this one, oh, I'm really pleased it went well for you on the test. This one I will go over now then. By the way, guys, if you are just joining me, you're very welcome. I am on my YouTube channel and I have it actually live streaming there for you. And you can also join my live Zoom lesson if you want to. Okay, so let's have a look at this. So you're told you have got your Hox gene here. So it's important that we use the information. So we've got a Hox gene. Oh, I'm sorry to hear that, Chidima. I'm sure it didn't go as bad as you think it has. One of the best things about when you come out of an exam is you actually forget what you wrote down, and then as people start talking, they tell you what they wrote. You didn't write the same stuff. You start freaking out, and actually, what's happened is you haven't written anything worse than anyone else. Um, you've just written it differently, so don't stress out. Um, Naveen, I'm really glad that that table process helped you a lot because honestly, now I'm like, oh, I feel like I could do any of zero dilutions. I probably get myself tripped up at some point, but at the moment, I'm on a roll. I'm positive with it. Right, back to this one. Sorry. So what we have here with this question is we have got the Hox genes. You're being told you've got 180 base pairs. So now we need to make sure that we're remembering what that means. So if I've got 180 base pairs, that therefore tells me that I have got 180 divided by 3, which tells me how many amino acids that I could have with the, I keep going to look for my calculator and it's not there. So base pairs, if I had it going A, A, T, T, C, C, G, T, A, when I talk about base pairs, I'm talking about A and T, A and T, T and A, T and A, G and C, G and C, C and G, A and T, T and A. Yeah. When I'm talking about my triplet code, there's 3, there's 3, there's 3. So if I've got 180 base pairs, and I know that there are 2 species with a homeobox sequence of 180 pairs, where 1.7% of those bases are different, and they want to know which of the following shows the number of amino acids that could be coded for with this. So if I understand that my triplet code, so I have got 108 bases. Should really buy myself a proper calculator and not rely on having my phone, but it's normally my phone that's there. So I've got 180 bases. If I divide that by 3, that tells me that I've got 60 amino acids. Okay. I can't believe I need to calculate that. Can you tell I'm tired? This is my 7th hour of teaching and I marked an exam paper today. So I'm literally like, ah. So base pairs or bases code for the amino acids. Well, the bases code for the amino acids, but remember the bases always come in pairs. So when they tell you you've got 180 base pairs, that's saying you've got 180 of these. So whether I'm saying I've got 180 base pairs or whether I'm saying I've got 180 bases, I'm saying the same thing because you know that one of them is the template strand and one of them is the part that actually codes for the antisense strand. So it's the complementary bases. Paul, do they include stop and start codons in this or not? Well, they do talk about stop and start codons, but they don't mention it when they're looking at this particular part. So they're saying there's that 180 that is the conserved part, which is your Hox gene, and any deviation from that could cause there to be problems, which could be lethal, which is what OCR want them to know. So in this case, because we know we have 180, we're told that 1.7% of the bases are different. You therefore would want to work out the difference. And basically, it's wanting you to realize that actually would be different in the 2 species. It could be a minimum of 0. So it could be a minimum of 0. And why could it be a minimum of 0? Because remember, you have your degenerate code. So that could be. Oh no, I just made it up, Annie. I half the time whenever I'm doing this, I am literally just making it up as I go. Sometimes it's a good thing that I make it up. Sometimes I trip myself up because I make it more complicated than, but yeah, I was just making up the AATTCCGTA. Any teacher who knows that off by heart, hat off to them because I don't remember any of that stuff. It's a funny thing, the things that you will remember and the things that you don't when it comes to biology. But anyway, so therefore now we have to decide would the maximum be 1 or be 3? And so therefore, again, trying to do this, if I look at my 180 and I divide that by my 1.7%, I therefore would get 3.06, which would mean, yeah, AUG. It's the only one you need to know when you start codon because it's methionine, right? That therefore would mean that 3 was my maximum and my minimum would be 0 because of that degenerate code. Right, someone asked for another question, which was in the same paper. Lynn, what was yours? 2019, paper 2, question 17, BII 17. And you want B, I, right, hold on, B, and you want I. Yeah, perfect. Is this the one you're looking at? So in this one, this question here, we are asked about gene therapy, and we're told that we have got genes that have been isolated from DNA. So that's important. They've already told us that the gene was isolated from the DNA. So for those of you who don't remember, your mRNA would need reverse transcriptase. This is where my reverse transcriptase. Reverse transcriptase to give you your complementary DNA. However, in this case, they've said that the gene was isolated from DNA. So therefore, I know that I'm using restriction endonucleases. So I've got restriction endonucleases here. I'm being told that we've got cystic fibrosis. I am being told, what else am I being told? Healthy individuals have a gene that codes for the channel protein found in the plasma membrane, and it's to do with the lining of the airways. People suffering from cystic fibrosis have 2 copies of a recessive allele, so the cells do not synthesize the correct channel protein. The allele that codes for the functioning of the CFTR protein can be inserted into the DNA. If it's going to be inserted, we need some form of a vector of the CF sufferers. The cell can then synthesize the correct CFTR and normal. Right. So you said BII. Oh, sorry. Did not see you there. Some attempts at gene therapy have resulted in changes of the functioning of the gene. Okay, explain how inserting a new gene could affect the function of other genes. So, don't worry, Paul was asking about start codons and he was doing the AUG, which is the start codon for methionine, which is why I said it was methionine. Oh, okay. I'll come back to that question in 1 second. I want to do this question first because it's actually related. So if I have, and I'm just going to make it up again, if I have my DNA sequence, so A, A, T, C, C, G, A, T, T, C, C, G, A. So let's say that's the sequence that I have. If I'm going to insert a gene, so let's say I'm going to insert the gene, and this gene is going to have A, C, C, G, G, A. Again, I'm just making this up. So this is 2019. We're now on paper 2, question 17, BII. So if I were to add this in, and I put this gene in, now what I would do is I would actually have, so I've got AAT T, C, C, G. I'm going to switch my pens to represent this. So I'm now going to add in my A, C, C, G, G, A. And now back to my red pen. And now I'm going to add in, oh crumbs, where did I get to? A, T, C, C, G, A, T, T, C, C, G, A. Okay, so if I have inserted the gene, then what you have got is knowing my triplet code. There's 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3. What I've now got is 1, 2, 3, 1, 2, 3, and so forth. So what I've actually done is I've done a frame shift. So because I've done a frame shift, that has therefore resulted in a different combination. And because I've got that frame shift, I'm altering the triplet codes. Before it went AAT, CCG, then ATT, whereas now I've got AAT, CCG, whereas now I've got TCC. So what you can end up doing is you can end up with this frame shift. And because you've got this frame shift, it's therefore had an effect on the amino acid sequence, therefore affecting the functioning of other genes potentially. Right. Amna, let me work out what you're trying to say here. So the base question, you don't get 3 bases for one codon equals 1 amino acid. Yes. So isn't it still 1, not 3? I don't think so. Why would that change 3 amino acids if the 3 bases are different? Okay, so hold on, let me go back up to that question a second. So when I did 180 and I divided it by my 100 and times it by my 1.7%, that told me that 3.03 of my base pairs, 3.03 of the base pairs were going to be different. So you're assuming, Amna, that those 3 bases are within the same triplet. But it could be that actually there are 3 different triplets that are affected. Does that make sense? Yeah, that's fine. That's the thing. That's the whole point and why I want to do these sessions for you, because figuring out those bits you don't get, that's what's going to make the difference of you getting it. And if I can't get it, if I can't explain it to you, it means I'm not really sure myself, which means I then will go away and think about it and try to figure out how to work it out. That other graph question, I'm not even bothering wasting the mental capacity on that one. I hate that question. It's done, dusted. They're not going to give me one like it again, so I'll just move on. But that's a difference because I don't have to sit the exam. So there are questions like zero dilutions that I know will come up again and again and again. And so I had a student who questioned one of my videos that I put out there, and then I started questioning myself. And then I was like, no, I'm wrong. No, I'm right. No, I'm wrong. And so I spent like 2.5 hours going over it and over and over. And I was like, oh, I'm so confused. And then Alan showed me this way that made it make sense. And now it's about the practicing. So if there's a question like that that just totally throws you off and you're like, I just don't get it, don't waste the paper. Don't waste the time, doing that question over and over again to get it. Just chalk it down to, I'm not going to get that question. Move on. It's 1 or 2 marks overall that I may lose, but let's nail all the other marks and that will get you the marking points. Could I go through an Edexcel paper as well? I can potentially go through an Edexcel paper if I can get access to it. Depends. I do Edexcel B. So if you can tell me which paper it is, then potentially. But let me just go back. Somebody asked a question on here. I don't know if you're still in here. I got a 6 marker about how organelles work together to make amylase with a specific shape. What could I have mentioned? How does organelles, right? So you're talking about amylase. Amylase is an enzyme that's going to be secreted outside of the cell. So the fact that it is going to be an extracellular enzyme, an extracellular protein, it's a protein that's going to be produced through transcription translation. That therefore means that it's going to go to the ribosomes which are on the endoplasmic reticulum, the vesicles from the RER to take it to the Golgi apparatus. The folding and modifying of it in the Golgi apparatus is then going to result in it being packaged into a vesicle to then be released by exocytosis. You're going to need the cytoskeleton to move the organelles. So therefore, to move the RER vesicle to the Golgi apparatus, to move the secretory vesicle from the Golgi apparatus to the cell membrane. Have I missed anything? The fact that you're talking about a protein, the protein having a specific shape, that's whether or not it's asking you about the fact it's an enzyme and therefore its tertiary structure. So the fact that the translation gives you the primary structure, the sequence of amino acids held together by the peptide bonds is then going to result in the secondary structure being the alpha helix or the beta pleated sheet. That therefore is going to continue with the tertiary structure, the interactions of the R groups, those interactions therefore resulting in a rounded globular shape that's specific. So amylase has got an active site which is specific for the breakdown of starch. So that's where I would have gone with all of that. Avira 21A for that paper, yes. So no, I don't have access to the 2024 paper for Edexcel, I'm afraid, because they keep the papers in lockdown unless you work for the schools where those papers are actually sat. And I work for a school where my students sat the 2024 paper, so I have access to that. So I don't have the 2024 paper, I'm afraid. Right, so what we doing 21A on this same paper? Let's have a look. Where is 21A? 20, 20. Oh, thumbs went too far. B. I like this question actually. Again, it's one of those ones when it's been a while since I've done the paper, but when I do see it again, I'm like, oh yeah, I like this paper. Okay, so when you say, Annie, just to, I'm going to ask this question in a second, but just when you say if it says describe, suggest, if you give 2 to 3 answers, it depends on how many marks it's worth. If it says describe something, that means say what you see, say what the graph is showing you, say the relationship between this variable and that variable. If it says suggest one of the things and you give 2 to 3 answers, now I get what you're saying. So if you give more than what is expected in the answer, if it says give us 1 and you give 2 or 3, we go by the first one. Unless it says ignore, then if you give a wrong answer in the first one, you're not going to get the marking point just because you've given us extras. So in that case, if it says do 1, I would say try and take the time to figure out what the 1 is. Okay, 21 AI, please not B. 21, yeah, AI, here we go. This one's an interesting one. I have to say, with this question, this is expecting you to use your GCSE knowledge as well when it comes to maths. This is a math skill question. So you are told in the question that you have got a number of phospholipid molecules in the outer surface of the lipoprotein. You are told that you're assuming the pattern of proteins and phospholipids continues across the whole surface of the lipoprotein. And you are told in the question that the surface area of a sphere is 4 pi r squared. Okay, so you've been given that information. So therefore, what you need to do is, first of all, count the number of phospholipids. So calculate the number of phospholipids. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21. So I know, because I've counted them, that I have 21 phospholipids. So I know I've got 21, which are going across my whole phospholipid at the moment. So then what I need to understand is 21 is the surface area of my sphere is 4 pi r squared. So then I need to work out, right, okay, well, what does that actually tell me? That tells me that I have got 21 phospholipids in the whole circumference. And this is where, when you look at, let me bring up, where is the thing here? Page, I want to say it's like 61. Oh, no, let's try that again. 61, there we go. They are expecting you, so yeah, it's page 62. On page 62, so go to page 62 of your specification. In there, you will be told information that you should know. So 2 pi r is how you work out your circumference. So if you know that your circumference is 2 pi r, therefore, what we would want to do is take our 21 phospholipids and divide that by our 2 pi to give us our r. So once we've worked out our r, then we need to substitute that in. How do you know it's 21? For the circumference, we know the circumference is 2 pi r, and I know that there are 21 because I counted. So if I'm going all the way around, I'm working out my circumference, and therefore 21 divided by 2 pi will give me what my r is. So if I do that on my calculator, that will give me, where's my calculator? Here we go. Clear. So 21 divided by 2 times pi, those brackets, equals, so r equals 3.342253, blah, 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 blah. Now, I never round until the last moment, just to be clear. I won't round, but I'm going to, just to make it easier on myself for writing. 3.34 is r. Well, now all I need to do is do, well, 4 pi r squared. So therefore, I need 4 times pi times 3.34 squared. No, we're not adding the proteins in it because we are told we've to assume the pattern of the proteins and the phospholipids continues all the way around. No, you don't need to protect and double the answer at all, because the phospholipids are within, each one is a phospholipid. It's not a phospholipid bilayer. Be careful, we're not looking at a membrane here. We're looking at a lipoprotein. We're looking at a structure that allows for the carrying of these fat molecules, because you know that lipids are not soluble. So I was going to say not soluble, insoluble is the word. You can tell it's getting late in the evening. So the lipids are insoluble. So the only way that they can be carried Absolutely, Chidima, is that it's through what's known as a Michelle. So if you're AQA, you learn about this more with OCR. They will throw this question in there because they are expecting you to just pull the information out from what's in front of you. So Amna, no, not Amna, Pritesh, don't worry about it if you're OCRA. A Michelle is basically just a way in which fats can be transported in from the digestive system into the lacteal system. Don't stress about that. It's not anything that you are going to be asked about. Yeah, basically. It depends because you will have heard of them slightly in GCSE, depending on the course that you were taught. But anyway, coming back to this one, if I then put those calculations in there, that would therefore give me a answer, which is 140.5. So yeah, they will throw in things that you have never come across before, which are biologically correct, but everything you will need to know is in the question to answer the question. So in this case, everything you needed to know was right there in front of you. You just had to look for the information. Although saying that, the equation for circumference is expected that you do know this. Yep, they love will and evaluate question. The reason they love an evaluate question is because that is going to help you to know what to do with module one, because this is module one assessment where they evaluate. It's assessing your ability to look at the science and to draw conclusions by evaluating whether something supports it or does not support it. Oh gosh, that's another one I absolutely hate. Veronica, no, it's not the IB diploma program. I used to teach the IB for many years. I was the head of biology for the IB at my previous school before I moved over to OCR biology. So no, but it's very similar. A lot of the content does overlap. The IB just have a bit more specificity in what you need to know, like Hershey and Chase and all that stuff. But yeah, a lot of the science is interlinked. Um, collagen, collagen is insoluble because it's a fibrous protein. Right, guys, I'm probably going to stay for about another 15 minutes and then I will go on. So I'm trying to find where there were other questions. How do we know the area equals the number of, because they've told you that you've to assume that the pattern of it is across the whole surface. So they gave that in the question with the caveat of assume it's the same everywhere, that it's always going to be 21, whichever way you were to look at it in this sphere. What would you say is the most important thing to be revising? What's the most important thing? The whole course. Oh, thank you so much. That's lovely to hear. I want to help you guys as much as I can. So obviously these sessions that I do offer, because someone jumped on here last night and was like, oh my God, that's so expensive. And we've got loads of people out there can do it a lot cheaper. These are what allow me to do these free sessions for you. They actually are what I make my living for, but they are planned. They are literally, I've planned them to the nth degree and I adapt as I always do, to what's in front of me. But I want to be able to give back for what it is with my skill set because I stress out about the fact that a lot of students say to me that their teachers haven't taught them something or their teachers were off sick or their teachers were on a course, and I get it because I've done it myself. And the thing is as well, the reason I want to do these sessions for you guys is also because there's a lot of teachers out there, newly qualified teacher, and I was one of them back when I did my postgrad in 2007. When I did that, I didn't know what I was talking about. I didn't know the science. I didn't know, I knew modules, but I didn't know all of it. And so therefore, I had to go and find videos to teach me so that I could teach my students. And I want my videos to be there, not just for you guys, but also to help your teachers who are new into the profession, who do not have the time to be spending the hours doing the research like I do, because actually I have years experience, but at the same time, there are bits that I have to learn. And so this is why I do this. Um, oh, you're very welcome. Right. Okay, Amno, wouldn't that give the total area of the phospholipid? So what we're saying is the surface area of the sphere is that. So when we talk about surface area, it's like basically taking a piece of string and wrapping it around a ball. Okay, all the way around the ball until the ball is completely covered. So if we know the surface of an area of a sphere is 4 times pi times r squared, and we know we can work out our r based on the circumference, this is where they're checking your math skills. So they are assessing, did you know that that would equal 21? Could you rearrange that equation? And then once you figured out r 3.34, can you then substitute that in? So the 21 is the one that goes around. And because the 21 goes around and we can assume it goes around everywhere, that is what is helping. And so I tend to visit, I do these sessions definitely on a Wednesday. Right now it's between a Monday and a Tuesday when I have time, because I've had to move because I do group tuitions now on a Tuesday. It means that depends if the girls are asleep or not in time, whether I do it on a Monday or a Tuesday. Do they repeat some pictures? Yes. Do they ask exactly the same questions? Not exactly the same, but there has been, I was quite surprised last year they did. They were a bit lazy in some of the questions that they wrote out, where they brought questions or graphs back in that they'd used in the past. Do I think that happened again? Will happen again? No, I think they'll be on it for this time. Should you focus on papers or going over content? Papers, and going over the content of the stuff you don't know. To be honest, not everybody will know the whole of Module 5. I make sure that I know it. I make sure my teachers have taught it, because that's how you can see the bigger picture. But what I realize is that's because I've got ADHD, so my brain works in a different way, and I can find the interlinking parts. And because of that is how I teach. And for some people, they can compartmentalize things and they can teach what they know and become experts in what they're teaching you. But if I don't know how this is going to go off in different ways, how they could ask the questions, I don't know the topic area. So that's why I taught myself everything, and I made my teachers, I was horrible to my teachers, I made them actually piggyback their lessons, so they had to know the whole thing, because I didn't know it myself when I first became the head of biology. And so when I became the head of biology, I made sure I knew it all, and then I made my teachers know it all as well. And it's about timing, though. The thing is, the difference is I don't teach Key Stage 3, Key Stage 4, and Key Stage 5 anymore. I've specialized in Key Stage 5. Yes, I can teach the others. Yes, I can do GCSE. Yes, I can do other things as well. But because I'm now specializing, my brain is 100% in the A level, and the students I tutor during the day, they are OCRA mainly. I've got one who's AQA, I've got one who's EdXL, I've got one who's IB, but the rest of them are all OCRA. So therefore, I have the ability to know OCRA inside out. And also, because I work with so many different students, I also see where you struggle. And so then I try and figure out ways to make it make sense. And every single one of you is different. So each session I have with all my students have different sessions, even though they could be the same topic area, just like my classes would be different. And that's one of the best things about having ADHD. I could jump from one thing to another without even thinking about it. But right, I've got 9 minutes left. So hold on one second. I need to. I'm being tested here. Right, so I'm going to jump over here and answer some questions for those of you who are in this. Right, here we go. Amna, you're very welcome. lecture most of the days and spent most of the nights either marking or preparing lectures. That's the thing. Unfortunately, in schools now, it's all the extra bits that you have to do that makes it very difficult for you. Now, I have gone through all the way up. I was director of student progress, and then my girls came along, and I realized I had to make a decision. I was either going to spend my 80, 90-hour weeks doing that and spending no time with my girls, or I was going to change my lifestyle. And I changed my lifestyle. And 2 years ago, I went self-employed, went private. I go in and help some schools when they need me, but I literally decided to do this all on my own. I do not get paid anywhere near as much as I did working in the schools, but the satisfaction I get from helping students is worth more than that. I would like to get paid a bit more, but I have to obviously do the right thing that means that I can be there for my girls, and that's the important thing. And also, I think about my girls potentially getting up to A level. They're 4 and 5 right now. No, they're not. They're 4 and 6. One's about to be 5. When they get up to A level, they can watch my videos and they can know it. So it's great. Right. Just to know from which countries are you all? And why would paper two before paper one? With regards to the you do the international A levels. Well, the thing is, it doesn't matter which paper they give you because actually they're assessing different parts. Um, right. Now you're just having chats in here. Go away and have your chats outside. I think maths overall is worth a third. Yeah, it is. Well, it's 15% of your overall grade for the maths. Yeah, no, it is. If you do OCR, it's about 15% of your overall grade, but it's how they ask it. When it comes to the maths and biology is 15%. When it comes to, and that's each paper is 15%, by the way. And then it comes to whether it's AO1, AO2, or AO3, and it's varies between each paper. Standard examination. Yes. What would I do if I didn't choose biology for A levels? I was going to do PE was actually my forte. I did PE, biology, maths, and English for my hires. And then when I started year 13 equivalent, I started in advanced biology, computer science, and German. But then what I did was I decided that school wasn't for me and I dropped out. And I didn't think, yeah, PE, physical education. I actually then went on, I took a year out and then I went to college and did a higher national certificate in leisure management. I thought I was going to own my own gym. And actually I realized it was the biology that I enjoyed the most out of it. I hated all the stuff to do with the politics, the legal documents, the business side of things. And so, yeah, then I left again. And then it was a year later after that, I then decided that I would go to university. And it was a toss of the coin, which decided it was going to be it was going to be biology over computer science. Because at that point, I thought computer science was what I was going to do. But yeah, there you go. Bit of a random one. Oh, well done. You passed all your PAGs. That's great. That's really good. And that means so you'll get your endorsement. So now the best thing to do is just double check the, oh, I don't know what I've just done there. I did not mean to do that. Sorry. I did not mean to do that. I literally just invite someone by mistake as I hit a wrong button. Guys, I am going to go. It's 5 to, yes, I do. I do online paid classes. I have at the moment a group tuition sessions going right now. I also have, but that one's sold out. What do I have at the moment? I have one-to-one slots available. And I also have a course coming up in May, if you're OCRA, in the May half term. And then I'm going to launch in September weekly group tuitions for year 12 and year 13. So I'm going to start doing that again from September. But I've got to get my current clients through to finish their exam, which is coming very soon, 5 weeks' time. I know, I bet you can't be can't wait to be done, Amna. It's scary, but it'll be a relief once it is done until you have to wait for those exam results to come in. So so my website is ukbiotutor.com. Can I spell? No. Tutor.com. If you look on there, you'll see my information. Chidima, you'll be absolutely fine with biology. Just take your time. You'll be absolutely fine, okay? Good luck. Good luck. And guys, I will be back on either Monday or Tuesday. You are very welcome.